Hello, listeners and viewers of the Retina Channel podcast. It's my utmost pleasure today to be here with Dr. Ursula Schmidt-Erfurth as in the University Hospital in Vienna. I'm Dr. Kuchernig, your host, as you know, and this is the first time actually recording an episode in someone's lab and research place. So this is a very unique episode. I'm very thankful to Dr. Schmidt-Erfurth to accept this invitation and have me in her office and as a pioneer in the field of artificial intelligence and many other things in retina. It's really an honor to be here, Professor. Thank you for having me.
Thank you for inviting me. Thank you for making this a reality TV show, because this is what medicine basically is. It's based on reality, it's facts, and we would like to improve the reality for our patients.
That's so true. So people in the field of retina know Professor Schmidt-Erfurth really well. You're one of the pillars in this field, but for those who don't know the background, you're one of the people who started PDT therapy when you were a fellow at Harvard, I think. That
is very true. Yes, that was one of my first brainchilds.
Yes.
Children, actually. So we did always, or my focus was mostly on retina from the beginning.
Yes.
With a priority in diagnostic imaging.
Yes.
And this diagnostic imaging consists of big data volumes, as we know that OCT is the type of imaging modality in vivo, non-invasive, that provides the highest density of pixels, the highest resolution imaging, and that of course is what AI loves most.
Yes. And I noticed that you've been in the field of AI since 2013. You established this lab, Optima Lab.
Yes, we did.
Which over 10 years ago. And now everyone knows about AI these days and learns about AI. And so you're well positioned to have this clinical output, this research output on AI. Mm-hmm. And hopefully we can get to talk about it today, if that's okay with you.
That's perfectly fine.
Okay.
So you mentioned that we have come a long way.
Yes.
Because AI is not trivial. AI, particularly in healthcare, has to be developed on ground truth so that we know that the things that we measure are really precise and objective, because there's a consequence to AI results being used in human patients. And we started a long time ago. Yes, indeed, we were pioneering, because we had these large amounts of data from having set up another 10 years earlier a reading center, which is an image analysis platform. Images are mostly digital, so we had large amounts, millions and millions of images, and we quickly understood that only AI can bring order and sense into those images, and this is when we started.
Very nice. So there are definitions about AI. There are different terms that people get confused. There's AI, artificial intelligence. There's machine learning. There's deep learning. Would you mind giving the listeners and the viewers some basic background about what these terms mean, each of them?
So machine learning is the beginning. This still needs annotation. It has the human in the loop. It depends on human experts. While deep learning is using convolutional neural networks of the brains, but technological brains, not human brains. And this is what is the base of most of our algorithms. They are using deep learning, and they identify patterns, they identify features, they do correlations, so that the devices, particularly in retina, that provide so many pixels, can be used by humans in the most efficient manner. So deep learning, I would say, is the keyword nowadays. Mm-hmm. And we are just bringing deep learning into the reach of the clinical community. Mm-hmm. We are working not just scientifically to develop algorithms, but we validated those algorithms. We made sure that they go through regulatory certification. So in Europe and in Australia and in Switzerland and Great Britain, those algorithms are already available. There are two different types of algorithms based on deep learning, which is the fluid monitor and the GA for geographic monitor, geographic atrophy monitor, and those are already tools. So we moved from pure algorithms, the idea or the theory, to the practical application by having CDSS, which is tools that facilitate diagnosis. They do not replace the human, but they give data, they give conclusions that a human doctor can use to make an informed decision.
Okay. So let's talk about specific examples of these applications. One obvious one that you mentioned in one of the recent articles you have is using AI to measure fluid volumes instead of central subfoveal thickness, for example. Would you mind talking about that, how that can be used and how you actually use it in how do you feed the OCT images to AI? And has it been used, is it used routinely now or is still in research?
So you already mentioned the path. So first you have to feed the images to the system. So in terms of fluid, you take an OCT. This can be any type of OCT. Let's focus on the spectralis OCT because this is where abundant data were available to train algorithms. So you can either switch or load up the image that you took on a spectralis OCT to the Epway in the instrument, or if you do not have a PAX platform that is Heidelberg related, you just use the Red Hub, which is an independent platform provided by Retinside. Retinside is the spin-off of our group who makes sure that the tools can be used outside. Once you have uploaded the image, the algorithm in the cloud will immediately work on it. If you're using the fluid monitor, you want to know where is the fluid located in terms of distribution. You want to identify intraretinal, subretinal fluid and PED because those are three different fluid-related biomarkers.
Yes.
And you want to know whether there's disease activity. That means that fluid is increasing. And you want to know whether therapy decreases fluid, so quantification comes in. By a mouse click and in real time, every clinician that is using this tool, which has MDR approval outside of the US and is an FDA approval in the US, but can be used whenever you notify your ethics, your IRB, that you're using this. Of course, you can use it. Then you will immediately at one glance see where the fluid is. Is it relevant fluid? We know that intraretinal fluid is most important, that this is the one that kills the photoreceptors. So intraretinal fluid, many tiny bubbles, have to be controlled tightly and it has to be treated. We have done a prospective clinical study comparing standard of care, clinicians treating any fluid to make sure nothing is missed, as compared to AI-based fluid detection. And we found that there are less treatments necessary if you're using the AI tool, that you can treat more patients faster because you do not have to waste your time by looking at fly-throughs. And with a lower number of treatments, you can have the same effect, and you can also identify patients that are non-responders based on the pattern of fluid.
Very nice.
So this is all what the doctor needs to know because anti-VEGF therapy is the most frequent therapy in all over medicine. It's the biggest budget drain in medicine in terms of investments, and we would like to make it work
more
perfectly
efficient.
fine.
So this is something already in use?
This is something that is already in use.
In Europe at least?
Yes.
Yeah, so the software reads off of Spectralis and analyzes it for better AI.
But we also provide it now for Cirrus, Triton and Maestro. This is all in the move.
Wonderful. Wonderful. So other than that, you also have other applications for AI. GA, exactly.
Why is GA so interesting? Because it's more frequent, 5 times more frequent than neovascular AMD. It's the major cause of irreversible visual loss. And the biomarkers are not as trivial as fluid, where you see bubbles or detachment. We talk here about subclinical biomarkers like ellipsoid zone loss.
Yes.
This is where the photoreceptors are located. And by using AI, deep learning based AI, that is identifying, visualizing, and measuring the thickness of the EZ layer, we could show that this is where disease activity is located in geographic atrophy, that you first have this EZ loss and then the RPE follows.
Yeah.
So this is what drives progression of disease. If you now want to treat in geographic atrophy, you treat patients that have disease activity. As the spectrum in geographic atrophy is so wide, some people don't move for years, others are losing vision in 6 months. This is what you can visualize by taking such an AI-based GA picture. And then when you treat, you also want to see whether the substance that has to be given lifelong really works. So again, you measure the effect on the photoreceptors, the EZ loss, and you know exactly whether the patient is benefiting.
So this is a change in really framework in how we think about GA. Has been on
Yes,
FAF
exactly.
images.
Yes,
FAF
it
images.
helps us to understand disease much better.
Yeah.
It helps us to, by a mouse click, non-invasively identify progressive GA as compared to resting GA.
Yeah. So looking at the photoreceptor health and EZ status versus end result of loss of RPE.
And do this early on, because we know that geographic atrophy starts outside of the fovea, and first visual acuity is perfectly fine until the fovea is wiped out.
Yeah, it's too
so
late.
we would like to see the loss in the photoreceptor anatomy before vision gets lost, and an OCT can do this easily.
Mm-hmm. And
we're also using this for screening, by the way.
For screening as well. And you have a line in your article saying OCT perhaps is a better tool for measuring GA than FAF.
It is a better tool because FAF only shows us where the RPE is already lost. But this is an end stage. It's almost a graveyard.
It's too late.
We need to see the level of disease activity, and that happens at the photoreceptor layer, EZ, the EZ layer, which is not visible to regular human examination.
And this is all by AI assistance, correct? You feed that information.
You feed the image. Basically, there's nothing magic about AI. It's also not so artificial. The way we use it based on OCT, it just looks on the pixels. It classifies each pixel, and it does so very fast, real time, within seconds. And it does so precisely that the error of deviation of such an GA algorithm is lower than 1 pixel. So the 1 pixel difference, if the device makes an error about 1 pixel, the deviation would be 4 microns. Our algorithm, as it has been trained over so many years so precisely, has a precision of 1.9 micrometer. This is below even the lowest pixel difference. So this is highly precise. And precision medicine is something that, particularly in retina, we would like to deliver.
Absolutely. Now this is fascinating. I think that would change how we look at GA and how we look at dry AMD. So is this also in clinical use? Has the software been developed enough for this as well?
This is in clinical use. Also the autonomous upload can be booked by a spin-off company which is called Retina, which is, by the way, working together with the device companies such as Topcon. And this is, you can also have access in any other country, as the procedure has shown to be safe and precise. But if you do not have regulatory approval, you just have to give your IRB a note, and then you can use it, and you will have the same measurements in real time. Just upload, independent of any PACS system that you have. So this is really available to everybody who is doing management of AMD, which we all do, because it's the most frequent disease.
Yeah, and I remember you had a sort of post hoc analysis of Oates and Derby, and you showed that if you look at GA this way, the effect of treatment is almost double. Is that right? Could you talk about that?
Why is the effect better? If you do a precise diagnosis of your baseline condition, because you identify patients that have disease activity. If there's a large area of EZ loss, which you can see as a green area on the image that the GA monitor provides, then you know that this is like the equivalent of fluid. In neovascular AMD, you treat when there's fluid. You would never think about treating in the absence of fluid and expect a benefit.
Yes.
The same thing, the fluid equivalent as a biomarker is EZ loss.
Yes.
That is shown by the AI algorithm. And if there's EZ loss, there's progression, and your treatment effect will be most impressive. So selecting the right patient, that is the most important matter in GA. And then the other thing is measuring function, measuring retinal.
That's the next area.
Visual acuity,
Yes.
precisely. In GA, it's not so trivial. Either you have full vision because the lesion is still outside the fovea,
Yes.
or the fovea is affected and your vision is very low. But there are many steps in between in the path of progressive vision loss. And this is where microperimetry comes in. You remember this tedious method where a lot of test points are placed in a macula which is moving. So we have revolutionized this. We just are bringing targeted microperimetry to the market together with iCare, the company that provides the Maya OCT. And in their Maya 3, an instrument that will come out at the end of the year, there is our targeted microperimetry based on AI again, where you have a standardized functional test that takes 4 minutes only, is fully automated, and places the test spots in a standardized manner in the area of EZ loss, so that we will be able to measure whether disease is progressing, vision loss is progressing, or whether we can stop it.
So this links everything together. This brings the sort of biomarker of EZ loss as something to measure for geographic atrophy.
Yes.
And links it using AI
to vision.
Functional outcome. Vision. Yeah.
Because vision is all that matters.
Yeah.
For physicians, and even more so for patients.
Yeah.
For clinical trials, for regulatory agencies, we know that the 2 substances approved in the US based on structure were not approved in other countries in the absence of functional evidence.
Yes.
But this fast AI-based test will allow to provide the functional benefit as well.
Wonderful, wonderful. Well, this is fascinating. Thank you so much. And you've been a pioneer in this field, not just this field, many fields of retina.
Well, pioneering never ends. We are still coming up with new things like the targeted microperimetry and other things that are a logical consequence of bringing more AI to a field that badly needs it and elegantly benefits from it, which is retina.
That's fantastic. Professor Schmidt-Erfurth, and I want to thank you for coming to the Retina Channel podcast, having me in your office, and all the contributions you've done to our field. You know, people like you push the field forward and teach the rest of us, and I'm thankful to you and your team for having led so many advancements. And I know we're going to get you in Canada for one of our Canadian Retinal Society meetings. Next few
With
years.
pleasure.
Yeah, it would be fantastic to have you in person over in Canada as well.
Thank you for your interest, and thank you for your knowledgeable questions.
Thank you very much. All right, friends, I hope you enjoyed my conversation with Dr. Ursula Schmidt-Erfurth in Vienna. This was a special in-person recording, and I personally learned a lot from her endeavors and all the work she's done, especially in the area of artificial intelligence and its applications to retina research. Our next episode will be with Dr. Christina Wang from Baylor College of Medicine in Houston, Texas. She is the Protocol Chair for DRCR Retina Protocol AO, which is the application of home OCT for monitoring of neovascular macular degeneration. So I look forward to that conversation with Dr. Wang. And until then, you guys take care, and I'll see you then.